Greetings everyone. Today we will discuss about study of ultrastructure and morphological classification of bacteria. This is the part of Pharmaceutical Microbiology Unit 1, 3rd semester, B.Pharm. So, two topics we will discuss. One is ultrastructure of the bacterial cell and morphological classification of the bacteria. So now in previous class we have discussed about prokaryotes and eukaryotes. So now these bacteria are unicellular organisms and they have variety of sizes, shape and envelope structure. So they are prokaryotic in nature. Prokaryotic means lower protista. These bacteria cells, they divide by binary fission, simple binary fission, and they do not have chlorophyll, and the size range for length is 0.5 to 15 micrometer and width is 0.2 to 2 micrometer. So this is bacteria. So prokaryotic in nature or lower protista, unicellular, don't have chloroform, and the dimension I told you. So now these bacterial cells, they have following organelles. Okay, so the minimal requirements for a cell to be prokaryote or bacteria. So these are the requirements. So what is there? There is cytoplasm, cell membrane, nucleoid, plasmid, cell wall, mesosome, and some surface appendages like flagella, fimbriae, pili, capsule. Some cells, however, have organelles like vacuole or some storage body, but these are not in true sense can be termed as true organelles. This is the diagram of a bacterial cell. So you can see the capsule, and this is the cell wall, this is the cell membrane, this part, this is the entire thing is cytoplasm, this is the nucleoids. Okay, so these things, ribosomes, so etc. are there. So individual organelles we will discuss now. So the cytoplasm, the content inside the cytoplasmic membrane is called cytoplasm. It has three types of zones, constituting zones. One is cytoplasmic area, which contains what? It contains granular substance like ribosome. And second part is chromatinic area. So this part, what is there? This is the nucleus, or this part is rich in DNA. Then third portion is, other than these things, the third portion, they contain dissolved material. And in the cytoplasm, the granules, ribosomes, they are 70S. Bacterial ribosomes are 70S. The sedimentation factor 70S. And the ribosome constitutes two subunits. One is small, another is large ribosomal subunit. So if we determine the sedimentation constant for each subunit, that is the larger is 50S and the smaller is 30S. So other than ribosome, there is other some granules, that is the metachromatic granules, they are called metachromatic granules or volutin granules, etc. So this is the cytoplasm. So cytoplasm, what is its function? It hold all the material, all the reaction takes place in the cytoplasm. And they act as a matrix of the entire cell. So now the cell membrane, though, or cytoplasmic membrane. So this is what? This is the outer covering of the, this is the outer covering of the, this is the cell membrane, this the first part, outer covering of the bacterial cell. So it is present beneath the cell wall and it is present above the cell. It consists of phospholipid bilayer, and in along with the phospholipids there are some proteins present. So the structure is like this. So these green colored things are, these are the phospholipid molecules. They are arranged like this. This is the polar part or hydrophilic part, and this is the non-polar part or hydrophobic part. So hydrophobic part, they face each other, and the hydrophilic part, that is, it is outside the cell. This is the inside the cell. This is the plasma membrane or cell membrane or cytoplasmic membrane. So the proteins are embedded. Some proteins are present through the entire cell membrane, and some are present in the surface, some are present in internal surface, and some are present like this. So this membrane speciality is, it is a semi-permeable membrane. That means what? It allows some molecule to pass through it. Both entry and exit, some molecule it allow to enter inside the cell. So that is very natural. Only nutrients, food material, etc., they can enter into the cell. Everything if enters into the cell unique characteristic of the cell will be, or environment of the cell will be destroyed. That is why semi-permeable. It allows some molecule, those things are required inside the cell, will enter into the, it will pass through the cell membrane. But undesired thing, toxic things, those things will not enter. So that is the cytoplasmic membrane. So what is the function of the cytoplasmic membrane? It gives the bacteria a shape, hold the material, and restrict the entry and exit of the chemicals. Next is nucleoid. Nucleoid or bacterial nucleus. So bacteria does not have a well-formed nucleus. So bacteria nucleus, instead of calling it nucleus, we call it what? We call it nucleoids. Nucleoids. So this nucleoid, it does not have any membrane. It does not, the nucleus is not membrane enclosed, and a thin fiber of double stranded DNA helix tightly coiled inside the cytoplasm. A single DNA, single circular DNA is present in the nucleoids. The DNA, this is very important, and it forms 2 to 3% of the bacterial dry weight. So now this nucleoid contain DNA which carry all the hereditary information of the bacteria. It help in the protein synthesis, it help in the synthesis of enzyme, and it also help in the bacterial metabolism. So by producing different types of enzymes and proteins. So that is the function of two things: they carry hereditary information from one bacteria to another bacteria, and second thing is it help in the protein and protein like substance like enzyme, that they help in the production of those things. So that was the nucleoids. The plasmid is also a genetic material. So definition wise, plasmids are extra chromosomal DNA, that is the piece of DNA present outside the nucleoids. Now this plasmid can travel from one bacteria to another. The copy of the plasmid can travel from one bacteria to another by a process called conjugation. So this the plasmid is very important because it is responsible for many bacterial capabilities, like its ability to resist an antibiotic, its capability to resist heat, its capability to resist chemicals. Those things are controlled by this plasmid. However, all bacterial cells may not contain plasmid. Even same bacterial species, some may have plasmid and say some may not have plasmid. So next is cell wall. Above the cytoplasmic membrane or cell membrane, the portion which is formed is called cell wall. So the nature of the cell wall determines whether a bacteria is gram positive or gram negative or acid fast bacteria. So now this it is the cell wall is a tough rigid structure surrounding the bacteria like a cell. So bacteria is there outside this cell membrane. So there is a rigid cell is there. So that is the tough and rigid cell, and it consists of 20 to 25% of the bacterial dry weight. And unlike cytoplasmic membrane, cell wall is porous, slightly elastic, and freely permeable. So you remember the cell membrane is not permeable, it is semi-permeable, it is freely permeable. And the cell wall is made up of peptidoglycan or murin. So peptidoglycan, so constituting material of cell wall is peptidoglycan or murin. So now peptidoglycan made of two things, naga and nama. So naga and nama. Naga means N-acetyl glucosamine and nama means N-acetyl muramic acid. So these are the this is dotted, this is the naga and this is the nama. So this is the naga nama. So this formation is like this. So one naga, after that nama. So this is forming a chain, another chain, another chain. So the chains are placed one side by side. Now these chains are joined together by pentaglycine bridge. This is the pentaglycine 5 amino acid pentaglycine bridge and peptide chain. These chains are interlinked to form the peptidoglycan layer. So the function I have already discussed. It gives rigidity and give protection, etc. So if we see gram positive and gram negative cell wall, there are some differences. So this is the gram positive, that is the cell membrane. Above this thing there is a cell wall. So and it is attached with the teichoic acid, uh, lipoteichoic acid. But gram negative, what is there? So this is the peptidoglycan cell wall, which is much more thinner. So another layer could be there outside the thing. Mesosome is a complex invagination of cytoplasmic membrane which act as an anchor during cell division. So if we go back to the cell diagram, so you see this is the cell cytoplasmic membrane. So some internal invagination, that is the cell membrane suddenly intrude into inside the cell. This is called mesosome. What it does, it help in the division of cell. Flagella are filamentous cytoplasmic appendages protruding through cell wall. So these are unbranched, long, and have a hair-like formation. Okay. Each flagellum, each flagellum singular is flagellum, and three parts. That is the filament. This is the filamentous part. Then hook part, and this is the root part. And what it does, it helps in the movement of the bacteria. Next is fimbriae. Fimbriae are thick filamentous appendages extruding from the cytoplasmic membrane. So these are the, this is flagella and this is fimbriae. They help in the bacterial attachment to a surface. Okay, say they have some spike. The bacteria have some spike-like formation. This is the fimbriae, and they are observed in both gram-positive and gram-negative bacteria. Pili, there are some other hair-like filamentous tubular appendages that is called pili. These are the pili, and they are longer than fimbriae but shorter than flagella, and present in gram-negative bacteria only. There are three types of pili: sex pili, common pili, and polycene pili. So what it does, it helps in the sexual reproduction of the bacteria. Some authors say this pili, fimbriae, and flagellum, they are similar formation. Depending upon the environmental condition, they are formed. Some authors say that, and some describe it as a separate organelles. Capsule, outside the cell wall, there is a jelly-like formation. So that is called capsule. So this is the jelly-like outer covering, that is called capsule. And what it constitute? What are the constituents? They are the polysaccharides or polypeptide or hyaluronic acid. These are the different constituent. Yes, polysaccharide, different types of polysaccharide could be present. Different types of polypeptide could be present. It may vary from bacteria to bacteria. Unlike peptidoglycan, it has different type of constituents. So what is the function? It gives protection. It enhances the virulency or disease causing ability of the microorganism. So this is what this is called morphological features or fine structure of bacteria. Now we will discuss about the morphological classification of the bacteria. So morphological classification means classification of bacteria according to their external characteristics like shape, size, color, presence of external organelles, etc. So as per the shape of the bacteria, there are following types of bacteria are there, like cocci. Cocci are spherical, round shaped like a ball. So these are the cocci, round shaped. So this is the cocci. Here two coccis are attached together. That's why it is diplococci. Then bacilli. Bacilli, they are rod shaped like a rod. You can see one rod, two rods, small rods, rod shaped bacteria. Spirilla, they have the shape of a S like this. This is the spirilla. And vibrio, that is the comma, like a comma. So comma, if you draw a comma, it is like this. So this is like a comma. And coccobacillus, that is the this is also bacillus, but the length is so less that it appear as a coccus. It appear as a cocci, so that is why they are called coccobacillus. Now this is the coccus. So these bacteria are spherical, ovoid, or generally round shape. Contrast to many bacilli shaped bacteria, most cocci bacteria do not have flagella and are non-motile. So another important point is cocci, they do not have any flagella. So all the cocci are motile, sorry, and uh, mm, now this during the cell division, the division can occur in different plane, and some cocci they occur, they form a chain string of beads or a chain of formation is like a chain. So you see there is a formation of a chain. This is the chain. So that is why they are called streptococcus. Diplococcus, as I told you, this is the two cocci are attached together, and then diplococci, and then the staphylococci, like a branch of grapes. This is the staphylococci. Okay, this is the like a branch of a grapes. Okay, so this is the cocci. So remember, spherical, oval, or round shaped. They do not have flagella. They are non-motile, and they can exist as two cells like diplococci, or like a chain of cells, that is called streptococci, or like a branch of grapes, that is called staphylococcus. Now bacillus, these are the straight rod shaped microorganisms. Almost all bacilli are gram positive rods. Diplobacilli, that means like the cocci here, two bacilli are attached from side by side, no, head to head. So not side by side, head to head they are attached. And if you see there, as you see like this, not by this side, that is a head to head. Diplobacilli, two bacilli are involved. Streptobacilli, likewise, that is the forming a chain of bacilli. Trichomes, so they also form a chain of bacilli, but attachment is more like this. This is the diplobacilli or streptobacilli trichomes. The attachment is more like this. More area are involved. Example is Bacillus subtilis. Spirilla, they are A-shaped bacteria. They are spirilla, they are helical, rigid bacteria. If it is not rigid, if it is flexible, then it is called spirochetes. Example of spirilla is Spirillum volutans, and spirochetes is Treponema pallidum. So this is the spirilla. Then Vibrio. Vibrio is a comma-shaped bacteria. That is bacteria with less than one complete twist. Okay, so and each Vibrio they have one flagellum. Due to the presence of this flagellum, they are actually partially bent. Example is Vibrio cholerae. Coccobacillus, as I told you, that is the rod-shaped bacteria, but length is so short that it look like a, that is length and width is same. So that is why they look like a cocci. And example are Haemophilus influenza, chlamydia, trachomitis are coccobacilli. So that was the according to the different shape. So now the flagella in different type of bacteria, flagella could be present. During the flagella distribution, there are many types of bacteria. Monotricha, that is one single here like flagella is present, that is called monotrichous. Okay, amphitrichous here, it is one single flagellum is present in both end, opposite end. If it is this end and there is that end, it is called amphitrichous flagella at both poles. Lophotrichous, the tuft or beard-like flagella at one pole. Peritrichous means the flagella is present all over the bacteria, all over from anywhere and everyone, the flagella will come out. Amphilophotricus, this is the Lophotricus. Amphilophotricus means, the term means both. This this side tuft of flagella and this side tuft of flagella is present. That is the tuft of flagella are present in both the ends. So that was the morphology. So we have discussed about the fine structure or ultrastructure of bacteria and morphological classification of bacteria. Hope you find it useful, and I thank you for watching.